So I wanted to compare the TRP operon and the LAC operon of a bacterial E. coli cell. So the TRP operon, it's good to note that it's usually on because TRP operon, it produces these genes here that create tryptophan. And tryptophan is an amino acid that we need to build proteins. So because we always need protein, this is, you can almost say it's always on, it's mostly on. And so even the name can kind of help you remember if you think of tryptophan as being a repressible operon, that means that it's typically not being repressed. It's typically being expressed. So that's one thing that's going to help me remember. So this part from the promoter to the end of these genes, this is called the operon. And over here, this guy it assists, but I don't think it's technically part of the operon, the regulatory gene. The regulatory gene makes these repressors. And what repressors are supposed to do, no matter if it's for the TRP operon or for the LAC operon, a repressor is supposed to repress or stop or block RNA polymerase from transcribing genes. So when there's just a little bit or an average amount of tryptophan floating around in the cell, this guy is on and he's making more. The RNA polymerase is transcribing these genes, and so that the end result can be that more tryptophan is being made. So what happens when there's too much tryptophan and it's just not using up enough of it is down here. So now we have all these extra tryptophans floating around, and the cell wants to conserve energy. So what will happen is, if this is called a repressor, the tryptophan itself is called a co-repressor, and it will go ahead and it'll sit inside of the inactive repressor. Once it sits in the inactive repressor, the repressor will change shape, and now it can fit and sit in the operator. And you can just see once it's sitting there that it's definitely building a wall blocking the RNA polymerase. It cannot keep going this way. It cannot express those genes. So once the tryptophan is used up, what will happen is the tryptophan that's sitting here inside of the active repressor will also get used up. And then the active repressor will float out of the unbind from the operator. And you'll go back to this situation where the repressor is inactive and the genes are being transcribed as usual. So we're going to go down to the LAC operon. So the LAC operon has the same machinery. The LAC operon is considered an inducible operon. So that can kind of help you remember that if it can be induced to turn on, usually it's turned off. So kind of up here, I drew the hamburger to get the idea across that we always need protein, but we don't always need lactose, and neither do bacteria. So since lactose isn't always around, the cell says, well, let's conserve energy. Let's not make these enzymes unless we really need to. And so that's what happens. On a normal basis, the repressor here will sit inside of the operator, and it will block the RNA polymerase. And the RNA polymerase will not be making these genes here. No transcription, no translation. But if glucose and lactose are both present, because the cell always has enzymes to break down glucose, which is a monosaccharide, so it's much easier to deal with, it still will not make these enzymes here. It still will not. This active repressor will still stay in the operator, and you still will not have any enzymes to break down that lactose. However, once the glucose is used up, then the gene will start transcribing those enzymes, and it will start making the enzymes needed, like beta-galactosidase, permease, and transacetylase, that will break down the lactose. So when lactose is present, it turns into this form somehow, I don't know the details on that, called allolactose. And in the case of an inducible operon, we don't have a co-repressor, because that is for the repressible operon. We have an inducer. So in this case, the inducer is the allolactose. So if normally the repressor has nothing sitting inside of it right here, and that means it's active, the inverse occurs. So when allolactose is moved around and placed inside of the inside of the active repressor, it makes it inactive. And when it makes it inactive, then it no longer fits inside of the operator. It gets removed. And once it's removed, well, look who's free now. It's the RNA polymerase. And now it can go across and it can start expressing this gene and making the enzymes to break down all of these lactoses. So the LAC operon is as needed, and the TRP operon is usually on. So those are just two key things to take away.